So here are the answers to the questions from the previous chapter. So why don't you pause the video, tick your answers off, and then go on to the next chapter, which is embryos, stem cells, and differentiation. So at fertilization, the gametes fuse. It's an important word, use the word fused. Now the fertilized egg now has two sets of chromosomes, one set from each parent, and it now will divide repeatedly by mitosis to form the embryo, which is the ball of cells. Now at an early stage, these cells in the embryo will start to specialize or differentiate. And this means that they can form the different tissues and organs that are needed for the fetus to develop. Plant cells can differentiate throughout life, unlike animal cells. Now you need to know about stem cells. These are undifferentiated cells. And what's special about them is that they have the ability to develop into any kind of cell, for example nerve cells. And they can therefore be used to treat paralysis. Now, it's very exciting. The developments in stem cells could have great potential in medical treatments. But the downside is that they are often obtained from discarded embryos, although they can also be obtained from the umbilical cord blood of newborn babies. So there are pros and cons for the use of stem cells in medical treatments. The advantages are that these treatments could possibly find cures for many serious diseases and illnesses that we have no cure for at the moment, and they can save lives. The embryos could be discarded from fertility treatments, unwanted embryos from IVF, and they may be discarded anyway. We could use cord blood stem cells, and this will avoid the need for embryonic cells to be used. The disadvantages, though, are that the use of these embryos will lead to the destruction of human embryos. So that's ethically very questionable. And these treatments are experimental or unproven so far, so we don't know yet whether they'll lead to any secondary problems. We could screen embryos for genetic disorders, which means that we can test embryos to see whether that individual is carrying the genes for any inherited illnesses. Screening can be done before or after implantation. So if a couple is undergoing IVF, when we have the embryos in a petri dish, those cells can be tested to see if the embryo is carrying any inherited disorders. They can also be tested during pregnancy, so if the woman is pregnant, then some cells can be withdrawn from the fetus or from the fluid surrounding the fetus, and those cells can be tested for inherited disorders. The trouble is, if the fetus is found to be affected, then those parents face a very difficult decision, because if they then know that they're carrying a child which will have a serious illness, they then have the difficult choice about whether to terminate the pregnancy or not, whether to have an abortion or not. So the advantages of embryo screening are that it avoids the necessity to give birth to a severely sick or disabled child. If the embryos are being screened before implantation, then they can just select the healthy embryo to implant. If it's a pregnancy that's been tested, then if the parents decide to continue with the pregnancy, then at least with the knowledge they can prepare for the arrival of a sick or a disabled child. There are disadvantages though. What happens to the embryo or the fetus that is found to have the disorder? Well, that individual may be discarded, and again there's this ethical problem of the destruction of human life. Okay, that's the end of that chapter. Here's some test questions to check your understanding.